Wrong one. Okay, I'm admitting, almost done admitting everybody. People keep joining in. Can you guys hear me?
Yes.
I can.
Okay. Are you all disappointed you don't get a snow day for online classes? I know you are. Sorry about that. But today we're going to kind of catch up a little bit and finish the odds and ends that we started before the diffusion and the movement of molecules in and out of cell membranes. And we didn't finish that chapter because we kind of were finishing up on all the chemistry stuff. And now we're also getting into the cell metabolism. So now we can kind of try to put the end part of that chapter with the diffusion of molecules and mitosis and the cell cycle and all the cell metabolism stuff together. So it's heavy on the cells, and then we go into tissues next. Okay. And I know it seems like we're a little bit behind, but we'll be catching up. By the way, everyone who has lab today, this has been what my understanding of the position of the school is on all this is that they canceled in person. So they canceled lab today, which I kind of anticipated was going to happen. So anything that was supposed to happen live and in person is not happening, but I have that old lab that should be helping you finish the lab material. A couple of people took the option and came in on Tuesday that we're supposed to be coming up today. The bone lab you can pretty much do on your own at home. It's kind of hard to get the feel for how the bones fit together. That's what you're supposed to be doing in person in the lab. I think because we've had to kind of truncate the labs and rush into the labs. Mostly I've been presenting the slides in lab and people kind of are looking a little bit at the bones, but I've actually been a little bit surprised at how little most everybody has become involved with the bones in lab. And I think it's just because we jumped right into the bone labs and are doing 2 of them at once. And it's too much material to present all at one time. If I had my druthers, I would rather have had half the class come for the first hour and a half, or half the class come for, and half the class come for the last hour and a half. It's hard to do it all at once. Those of you who've showed up can probably speak to that. But you know, it is what it is. It's what they organized. There's not much I can do about it. So at some point, if some of you who've missed out on the bone experience, when the weather gets better and you want to have a bone experience, we can probably try to arrange like maybe a makeup lab day or something. I can probably see if I can get in there and maybe even like on a Saturday or something if it's more convenient for you guys. But I don't want to make any promises on that. I'll check into that. Okay.
I had a quick question. Are you going to go over the first PBL today?
Oh, the first PBL?
Yeah, are you gonna?
Because it's supposed to start on the 4th?
Yeah.
Yeah, I think I'm going to delay the start of that until next week because with the snow and everything, it's probably not a good time to start it. I thought that we would be over with the whole lab thing by then. So let's change the date of the PBL. Thanks for bringing that up until next week. Are any of you disappointed on that?
Is it going to be
Really?
No.
Is it going to be just the first 4 chapters that we?
Yes, it'll
gone
just be
over?
the first. It'll just be the first 4 chapters, and that way, we can talk a little bit more about it on Tuesday, and I'll open it up then Tuesday night. Does that sound good? And still give you like 5 days to finish it.
Yeah, that's good.
That sound better? Because otherwise, I think we're too rushed with starting a week late, having a snow day, yada yada. You know,
I've got a quick question about the Connect. I don't know if anyone else has had this problem.
on some of those slides where it's like, what is the highlighted region? I don't know if it's because I am partially colorblind. Like some of those slides, I can't differentiate what's highlighted. And I'm
Oh,
just like I'm looking at the pelvic bone and where it connects to the hip bone, and it's like, what's highlighted? And I'm like, oh my God, this looks like nothing is highlighted. And all I can see
is
is the
this Aaron?
picture. Yep,
Who's
this is
talking?
Aaron. This
Yeah, I
is
thought it
Aaron.
was Aaron. I thought it was your voice. See,
Some of them were really tiny and hard to see.
okay. So definitely let me be aware of that. And if it's a problem, and it
Because
affects.
this is my second time taking this class, and the first time I took it was when I learned that I was colorblind by going through those booklets
Uh-huh.
during the eye portion for the eye test to see if you can see certain colors.
Right.
And
right.
so like some of these slides, I'm like, this whole picture looks exactly like nothing is highlighted. Some
Okay.
of them I can see, some of them I can't see anything highlighted. So
All right. So if that's affecting your grade, I'm glad to be aware of it because we can fix that. And I can kind of almost give you your own separate thing of bones that I've highlighted and let you kind of practice with that, Aaron.
okay, that would be very helpful.
And I'll make it like a real obvious kind of dark contrast area from some slides. All right.
Thank you. I
Yep,
appreciate
so I'll
that.
send something like that.
Aaron,
I'll even put it out there for everybody to practice too. Okay, so I'll work on that this afternoon. I'll set up some testing up of the bones. All right.
on some of them, Aaron, if you roll your mouse over the bones, it'll pop up where the highlighted region is and it'll say like, name this region. This
Okay, I appreciate that. I'm going to give that a try then today.
Okay,
Thank
and
you.
I'll reset it for you trying again if you've already turned it in, so that you get a second try on it.
Okay.
That's bad if that's not allowing for the potential for colorblind, because we always have somebody who's kind of colorblind in the class. Just by the nature of being male, it's more common. So
Unfortunately.
it is unfortunate, although worse deficits can be found. I had actually my neighbor across the street, speaking of deficits that we all can have, they just became grandparents, and I was asking her how the first visit with the new baby went. They drove out to New Jersey to see the new baby, and they all just discovered the baby is deaf. And it turned out, oddly enough, as we get into all this, the baby was born with a vestibular nerve, but not an auditory nerve. So later on, you'll learn that they're kind of considered as part of the same nerve, the vestibulocochlear nerve, nerve 8 of these nerves that come off the brain. So it has the one for balance and control, but not for hearing. And I didn't want to say this because they wanted to investigate the idea of getting a cochlear implant. And the cochlear implant attaches into the auditory nerve. And I didn't feel like it was my position to be the one to tell them the news that this isn't going to happen for them, because this baby is going to end up being deaf. There's no nerve to take the message from the implant to the brain. So it's going to be a very sad day for them coming up. But it's where we can put, anatomy that you haven't quite yet learned into practice here. However, the fortunate thing for this baby is that the great grandparents were deaf. So the grandparents already know sign language, as does the mother. So they will be able to start signing early on with this child. So he shouldn't have any deficits. All right, so the movements in and out of the cell we went over, and a lot of people have said they're still having some issues with this. Maybe you have fewer issues with it after you did the connect work, but let's kind of review a little bit about this and then go on into the other material. So this is old material for the first 4 or 5 slides. All right. So when we're going to move things in and out of the cell, we have our cell's semi-permeable membrane. And remember, we just built up all the cell membrane with having our hydrophilic, water-loving phosphate heads facing both the inside of the cell and the outside of the cell. And then we have our hydrophobic fatty acid tails kind of linked together, and they form the hydrophobic part of the cell membrane that keeps everything excluded from moving in and out of the cell. So a lot of times, as you'll see when we get into this, we'll have transporters, membrane proteins that are integrated throughout the membrane that might have a channel in them that help us move materials in and out of the cell selectively. Now, some things can just diffuse across the membrane, and this is going to usually be small molecules, and they move passively, okay? So it doesn't require ATP. It's not going to require usually fancy transporters of any sort. And diffusion is that things are going to move just down their concentration gradient from an area of high concentration to an area of low concentration. You know, when I gave the example before of a garden hose, you turn on the spigot and the water finally moves down the hose from the area of high concentration at your house in the spigot where you turn it on. The thing that just causes the movement is really going to be diffusion. It just moves down the hose to where there is no water, okay? All right, so that's diffusion, moving from where there's a lot of molecules of one type to where there is little to nothing, all right? Examples of diffusion would be, you know, like the movement of carbon dioxide in and out of the cellular membrane. Filtration itself is a type of diffusion. You move molecules through a filter from above the filter where there's an area of high concentration to below. That is somewhat, of course, assisted, you know, in a laboratory situation where you're filtering something by gravity, okay? By gravity helps move that through, and even the atmospheric pressure above a filter sometimes. All right, so physiological processes that require ATP usage or this expenditure of energy are what we call active processes. So active processes will require us to, you know, use a little of our ATP, and oftentimes it'll require some transporter, but you'll see these are all active processes such as endocytosis, which we kind of think of as cell drinking, exocytosis, cell eating, and transcytosis, moving substances across from one face of the cell membrane, move it across the cell, and usually expunging it out the other face, okay? So we'll look at all these. All right, so diffusion, making sure that we understand this. When things diffuse, you have a high concentration, like we dumped this whole block of salt or a giant sugar cube or something here in water. So you have a big lump of it. It's all concentrated in one area. There's no salt out here. All right. Now we learned why salt dissolves in water, but instead of all the salt kind of staying in one area as it gets solvated, all these molecules are in constant motion, and the entropy, okay, entropy is sort of the tendency for things in the universe to move to maximum chaos. Entropy is going to make these molecules all want to move as far apart from each other as possible in their confined region right here. Just like when you spray perfume or air freshener or something at one spot of the room, all the molecules, all those odorants will move across the room, you know, till they kind of equalize their distance from each other within the confined spaces of the room. Is anyone having trouble understanding that? Okay.
I don't think so.
Okay, so the molecules are always in constant motion. Gas molecules, liquid molecules, especially in constant motion, gas molecules in motion a lot faster than liquid molecules. As we cool things down, make them a liquid, they have a little bit less motion. Solid, of course, has very, very limited motion. Okay, it's really the idea that we start making those atoms motionless in the molecules in which they are. All right. So only substances that are permeable to the membrane, that can get in and out and in and out with no, problem, no hindrance, are going to be substances that diffuse passively through the membrane, not requiring energy. All right, here again, we see we put a solute molecule, and then all this blue. This is a little confusing sometimes looking at this beaker because it's drawn in with all this liquid water. And then on top of that, they draw in the water molecules. So what's all this liquid behind it? Are they not water molecules? It's a little confusing. I can understand why some of you are confused by it. So you just have to imagine all the liquid water is blue molecules. There's a little bit less liquid over on this side because you have so much of this red solute molecule. And we're going to say this membrane right here is magically permeable to both water and to the red solute. So everything is going to equalize. Water molecules, which are high in concentration here, are going to move over to side A. So we're going to see blue molecules coming over here. And since these red molecules are permeable to side B, they're going to move over in this direction from their area of high concentration to where there is low concentration, because there is none. So the red molecules are suddenly going to equalize out over here, blue water molecules over here. And on this picture, you see they're equivalent. Okay, they've each moved to both sides. All right. Usually we talk in terms of diffusion of either the solute or the solvent, in this case being water. When we talk about the diffusion of water, we give it a special name, and that is osmosis, the movement of water across the membrane. All right, so diffusion through the cell membrane is typically going to happen through ion channels or transporters. In my microbiology class, I want somebody asked the question, so I want to make sure you understand that this is what's called an integral membrane protein right here. It spans through the entire phospholipid bilayer. Okay, so and it has its channel in here where we're going to let the molecules through, and it can kind of shift the opening of the channel. That allows for that selective permeability. If we were to have a molecule or a protein that kind of just, let's say it's stuck partway through, so maybe its face was facing the internal side of the membrane, but didn't stick out to the other side, it would be an embedded protein, but it wouldn't be integral. It wouldn't be considered through the whole face of the membrane. And you do have proteins that are just embedded in membranes too, okay? Not as integral, and certainly not all of them will function as channels, all right? So if we look at this channel, we're gonna say substances like this, okay, will be able to move through the channel. The channel is specifically for usually only that molecule, okay? And it's passive. There's no ATP that's expended. It's just that the channel kind of shifts its shape as the molecule is moving through the channel, all right, over to the other side.
So just so that I understand,
Mhm.
if we're talking about facilitated in terms of cellular, which we're talking about, we're always, that means that we're saying there is a protein carrier that makes this happen.
Right.
Okay.
Right. A protein carrier is shifting it through. Okay. All right. All right. So the special case of diffusion of water is called osmosis. So here again, we have our selectively permeable membrane, and it's permeable for, once again, the blue water molecules here. This is what's going to be able to move across in this direction or move across in the other direction. This other one, and I'm outlining these right now as I'm speaking because Aaron, you just said you're colorblind.
Hey, thank
So,
you.
Yep. So these big molecules up here, like this and this, these cannot move. Okay. All right. So what you see is on this side, we have the water that's able to move across, and on this side, the water able to move across. But because we have so many of these big giant protein molecules over on this side, the water is mostly going to move in this direction, trying to make those protein molecules farther and farther apart. Okay, so that is the diffusion of water. You want to make the concentration of water on this side of the membrane equal to this side. And that concentration is affected by how much of that bigger protein molecule is dissolved in the water, all right, or is solubilized in the water. All right, so where we always classically talk about this is the osmotic pressure that comes with in a red blood cell. But this osmotic pressure is faced by all kinds of cells. It's just easy to watch with the red blood cell, all right? So we're going to have the concentration of water on the outside of the cell and the concentration of water on the inside of the cell. Now on the inside of this red blood cell, there will be water, but there will be a lot of proteins in there, things like hemoglobin, okay? Other cellular proteins, other cellular solvents. Of course, on the outside of the cell, primarily the plasma that this red blood cell is floating around in is going to have also some proteins like albumin or some salts like saline, some sugars, you know, things like that that are in the plasma. And so the different solute concentrations, even though they're different substances, can make the relative water concentration on the inside of the cell and on the outside of the cell relatively the same. And when they're relatively the same, there's no net movement of the water outside of the cell. There's no net movement of water inside of the cell. Now I'm saying net movement because of course, water's kind of always moving a little bit in, but when it's moving out at an equal rate, the net overall change is zero. And this is what we call isotonic, okay? Now, if we put the red blood cell in a solution, maybe we take it out of plasma. Our plasma is usually isotonic. This is why when you go to the emergency room and the first thing they do is they hook you up on an IV, is they want to hook you up on 0.9% saline solution, because that is isotonic with your blood, your plasma, okay? It's about 240 millimolars of sodium chloride in there. All right, so the osmotic pressure is the same. It's not going to make your cells fluff up. It's not going to make your cells shrink up. It's going to keep your cells, you know, just exactly the same. If we were to take your cells and put them in a hypertonic solution, okay, like lots of sugar water or lots of or high concentration of saline, let's say instead of 9%, 0.9% saline, we decided to put you in a 1.5% saline solution, higher concentration. Well, then the water would want to run out of your cell to try to equalize the outside concentration of that sodium solution that you put it in, that's a higher concentration. Try to, you know, bring it down from 1.5% to 1% to getting down toward that 0.9%. Now, of course, if the outside water or saline solution isn't high enough volume, you know, all the water would end up leaving the cell, right? Because it's never going to be able to equilibrate with the whole outside of a high concentration and high volume of higher concentration solute. And that's when we say the cell crenates or shrivels up, okay? Crenate. All right, now if we put the cell out here in pure water, which has no solutes, we call that oh, by the way, that's called hypertonic, okay? When the outside concentration has lots and lots of sodium or other solutes in there, lower concentration of water. Here, when we put the cell in a high concentration of water, let's just even call it pure water, lots of water, no solutes, okay? No solutes. So this means it's a hypotonic solution. There's more solutes inside of the cell, all right? Less water inside of the cell. So all this water is then going to want to rush into the cell. And because there's always solutes inside of the cell, and if there's no solutes out here, this is always going to be less concentration. So the water will keep rushing in until the cell actually ends up bursting because you can't stop the water rushing in. And that's hypotonic, okay? All right, so why do we care about all this concentration and the example of filtration? Because we're going to filter lots of things across our semi-permeable membranes. And one of the places we can look at it is with the blood and the red blood cells moving through our blood vessels. Our blood vessels work as giant filters, all right? So we have lots of solutes, you know, blood cells, saline, the plasma proteins, all running through our veins and arteries, all right? Now, when we have high pressure, like blood pressure, okay, it's going to help, like I said, the filtration of gravity. And if we wanted to, you know, put pressure on our filter system to kind of push, you know, things through our filter. Sometimes you've been in chemistry lab where you put the pressure from below, like you put an airlock hose kind of putting pressure to suck things through your filter. I'm sure you've done that in your high school chemistry lab. You have one of those Erlenmeyer flasks that has the little hose suction coming out of it. That helps move everything through the filter a lot faster with a lot greater efficiency. And all your blood is under pressure with the pressure coming that your heart generates through all your vasculature. And that's going to help push fluids and solutes that can pass through the blood capillary wall outside, out of your vessels and into the tissue fluid that surrounds your blood vessels, okay? This again is a passive process. There's no ATP required to do this, and you have the assist of the heart's blood pressure, forcing things through the slits in between the cells, okay? In between the cells. All right, so then also at the cells, sometimes we're going to have active transport. Here we're going to have the movement of things across the cell membrane from high concentration still to low concentration, but we're also going to have ATP being expended to move things in the opposite direction, from low concentration to high concentration. And that's when we're going to actively pump things, say for instance, out of the cell. All right. Now, normally in our cells, okay, this blue side of the membrane over here is the extracellular side, extracellular or outside the cell, extracellular. And this is the intracellular side. We might as well start learning this right now, intracellular. On the outside of the cell, usually we have a high concentration of sodium, okay? High concentration of sodium. Inside the cell, we have a low, so I'll try to write it a little smaller, a small concentration of sodium. All right, outside of the cell, we typically have a low concentration of potassium. And inside of the cell, we tend to have a high concentration of potassium. And often we're going to have to maintain, we want to maintain our potassium concentration in our cell at a specific value and our sodium concentration inside the cell at a specific value. Same thing with the outside. So if potassium starts building up in the cell, we're going to want to pump it out. And same thing with sodium. If it's not high enough in here, we're going to want to pump it in. And what we'll find is our membranes are also a little bit leaky to potassium and sodium. So a little bit of potassium is always kind of leaking out the cell, and a little bit of sodium is always leaking in. And we have pumps that are going to try to keep us at the normal concentrations, all right, of sodium and potassium. So we'll see, we'll have a little membrane channel where we can get our carrier protein, say, transporting some of this sodium inside the cell, okay? And we'll also transport potassium outside the cell. But we have to use ATP, and you'll usually see it kind of written like this, because we'll usually use a little bit of ATP, in which we use that high phosphate bond that we talked about to drive the reaction. So ATP burns off one of its phosphates and becomes ADP. Now, sometimes, like I said, when we're doing all this and using the cellular energy, the carrier protein, in this case, we move something from the inside to the outside, it changes shape. Okay, so it was open right here when it was taking this potassium on the way through. So let me take that off. We'll have it going that way. When we move the potassium through here, on this face, it was open. What the ATP does is it helps change that conformation of that carrier protein to then spit this potassium over to the other side. All right. And that required some energy. All right, so let's look at some specific cases of active transport. So in endocytosis, we're going to move substances into the cell, and we're going to use a vesicle to do it. This is for substances that are too large to fit through something like a little tiny channel in a carrier protein. This is when we are going to be moving kind of things in bulk. Okay, so this is kind of like the Costco. All right, this is the Costco of a cell movement of substances. All right, so we have three basic types of endocytosis. Pinocytosis, cell drinking. Student last time told me she remembered it by Pinot Grigio. Whatever helps you. Okay, so cell drinking, Pinot. Phagocytosis or phagocytosis is cell eating. Okay, so phago, whenever you see that FAG, phagocytes are cells that engulf things. All right, so this is going to be taking in our solid particles. And then there's receptor mediated endocytosis. So we're going to use receptors on the outside of the cell to grab onto substances, and then they'll be concentrated at an area of the membrane, and we'll just take the whole membrane in. Yes, Nassau. How do I pronounce your name? Nassau?
Yeah, that's fine.
That's fine, or is that
fine.
sort of fine, or is it right?
Nassau.
Nassau. Okay.
Yeah.
All right.
So
I had a student I had for 2 classes, and her name was Elise, spelled ALYCE, and I called her Alice, and she never corrected me the whole 2 years until one day I ran into her in the grocery store, and I was like, oh, and I called her by the wrong name. And she says, that's okay. You and my grandma, you two are allowed to call me by the wrong name, but I don't want to call you by the wrong name.
Well, there you go. Thank you. Oh, okay. So what's the difference between receptor mediated endocytosis and pinocytosis?
It's really hard. Okay. So in pinocytosis, they're just taking it in liquids.
Right.
And I actually kind of think they're really somewhat the same thing, because I find it almost hard to believe that we're going to just take in a lot of liquid substances without really having receptors for those liquids. I think eventually we're going to discover that pinocytosis and phagocytosis are really a type of receptor mediated endocytosis. So the idea is that just there's water sitting outside of the cell membrane, and the cell just somehow folds its membrane around it, and pinches that water or that food in. But I think where you're going is you're wondering, like, why would it do that? And this is actually, you know, an area of actual investigative research, as I think we're finding more and more all of this. There's going to be receptors that grab onto that water and those solid substances and bring them in. So if you kind of even want to think of phagocytosis and pinocytosis as a type of receptor mediated endocytosis, I don't think you'd be wrong. All right. But we still separate it, for this is the world of the unknown as to what triggers the cell to move this stuff in bulk.
Yeah. What it reminds me of is sort of like a parasite.
It is. It's very pure. And, you know, and when you get into microbiology, that's kind of how it works with parasitizing something else, is taking it in kind of pinocytosis or phagocytosis. Yeah. So actually let's go on here on endocytosis when we're seeing it and when differentiating it with the receptor mediated endocytosis. So the idea here on an endocytosis is that somewhere here at the cell membrane, you take things in from the outside. You know, we would have, you know, a whole bunch of particles out here, and this area of the membrane is going to come up and kind of move around it, and it's going to engulf it and bring it in here into an endocytotic vesicle. And then within, once you phagocytose something, I should have drawn that out over here. So here's the thing that we took in. This would be like this stuff that we took in right here. And you have your lysosomes that are full of digestive enzymes. And somehow this vesicle moves toward the lysosome, and the lysosome moves toward the vesicle, and their membranes fuse together. So now your digestive enzymes can encounter that big particle that you brought in, and they can digest it, and it can move whatever it wanted out of that, you know, digestive, like let's say it needed some protein things, protein particles, some amino acids to help build the cellular proteins. It can also move toward the membrane here with what's left that the cell doesn't need, and it's going to expunge all the residue, all the waste product out of the cell. Okay, so that's that whole process. When you have receptor mediated endocytosis, we see the receptors lining up over here, and they're specific for these sort of pinkish particles, Aaron, that are triangular shaped. And the receptors grab onto this. And then you see in this area, this is actually, if we want to get really in depth, there's this substance called clathrin, and they form these what are called clathrin coated pits around here, where you have this high concentration of the receptors. And they form these coated pits, and then you bring in, and it kind of triggers that vesicle to form and bring in the endocytosed substances. You know, now, why would pinocytosis and phagocytosis, it's not supposed to have these receptors grabbing onto things? I think it's just because we probably haven't found and identified those receptors yet. That's probably what it's going to turn out to be. All right. Exocytosis is that, cell vomiting. Okay. Cell vomiting, we're going to expunge the waste process back out. So if phagocytosis was the cell eating, bringing it in, exocytosis is cell bulimia or something. It's getting rid of all that extra waste product, which is what we did right here. So this would be the exocytosis portion. This would be the endocytosis portion, or phagocytosis if it's solid. Okay. Examples of exocytosis we'll have is when we get to nerve cells. So putting things all together, you had your DNA in the nucleus. Your DNA gets opened up by we have RNA polymerase jump on there to make the messenger RNA. The messenger RNA travels out of the nuclear pores and goes over to the endoplasmic reticulum here, gets on those ribosomes, right? And those ribosomes translated into protein, so that suddenly we have all these proteins that get modified in the Golgi apparatus over here. Okay. And so now we have a completed protein, and our completed protein that's in this vesicle kind of moves across to the cell membrane, and it gets expunged out of the cell membrane. As you'll learn when we get to the nervous system, all this happens down the nerve. You guys have probably seen nerves kind of drawn like this before, where you have the cell body up here, and then you have all these dendrites coming out at the end of it. I'm trying to incorporate this as being the cell body up here. And we'll move the neurotransmitters down the whole nerve to the nerve endings. And all those neurotransmitters will end up getting expunged out into what we call the synapse area of the nerve. And then there will be receptors over at the muscle cell waiting to grab on to all these neurotransmitters that are being released by the nerve, okay, as they go across the synapse. If you didn't really get all that because you never studied nerves before, don't worry, we'll get into it when we get to the nerves. Transcytosis is kind of a combination of an endocytosis and an exocytosis. You're going to have a cell usually that has one face in the body. This is going to happen a lot at the small intestine, where we have a lot of microvilli. Okay, these are just little plasma extensions to increase the surface area. So in your small intestine, we're trying to pick up all the foodstuffs. So on one face, we got some food molecule, let's say it's an amino acid, and it's going to get endocytosed at the apex of the cell. The apex of the cell is facing the lumen of your gut, right? Where all your food is passing through your food tube, your stomach into your small intestine. So it's going to get endocytosed at the apical end of the cell. It's going to travel through the cell, and then you're going to have things like your blood vessels running below the cell at the other side of it. And we want to get all those food particles into our blood, let's say the amino acids. So we're going to have that food particle in here, and it's going to get exocytosed or expunged out so it can move into your blood and maybe like move through your blood to go to your liver. Okay, that's why transcytosis becomes important. Okay, so here you see a better drawing of it. Transcytosis, the example they have here is kind of a microbial example. So in a vaginal cell, we have HIV infected cells. All right. Anybody know what cells take up HIV? The cells are your CD4 helper cells, right? CD4 helper lymphocytes. So these are all infected CD4 lymphocytes, and they get taken up by the vaginal or anal cells, and HIV gets endocytosed at the apex facing the vaginal canal or the anal canal, and then it gets exocytosed, and it's really probably going to be moving through the blood supply that's underlying all those cells. Okay, so then you have this HIV in the white blood cells, the CD4 helper cells moving throughout here. Okay, and that's how HIV gets taken up in our bodies. All right. All right. So now we're going to move into the cell cycle. Okay, which you guys have all probably learned before. So your cell cycle is basically how the cell lives, reproduces into new cells, and then how those live and then reproduce, and so forth and so forth. Okay, so it's basically the life cycle of the cell. And our stages are divided between interphase, okay, which is where we're going to copy our DNA. Then we're going to have mitosis. This is where the cell splits into two new cells, cell division. Okay, but this is specifically division of the nucleus. And that's followed by cytokinesis, division of all the cytoplasm and all the rest of the cell. So you kind of think of interphase, and then you think of mitosis and cytokinesis as together. All right. So interphase is going to be all this big phase of the cell over here. All right. And then mitosis, we'll see it's divided into its separate nuclear division phases. And then that's followed by cytokinesis over here. All right. We'll also go through these important phases here that are restriction checkpoints. These are really important for determining whether we've replicated our DNA. And I should have that whole interface thing drawn all around here. This restriction checkpoint we're going to see is really important for checking DNA and making sure the DNA that we've supposedly replicated has replicated with enough fidelity that the cell can go on, that there haven't been mistakes that might be mistakes that are things like cancer, okay? And you can see sometimes those restriction checkpoints fail, but usually they work for us. All right. So mitosis is going to be division of the nucleus. Cytokinesis happens relatively at the same time a little bit, but is a little bit delayed. We've finished replicating all the, dividing all the DNA in the nucleus, and then all the rest of the cytoplasm kind of drags along a little bit after. Now, mitosis is going to be divided into all these phases. PMAT, P-M-A-T is how you want to remember it, okay? Usually, like anaphase and metaphase are really easy to identify. Prophase and late prophase gets to looking like metaphase. Telophase, it's a little bit harder to identify, but we'll look at the stages. So in prophase is where all that chromatin is going to condense up and form chromosomes. And remember, in the nucleus, this is just the nucleus. We had the nucleolus. The nucleolus is not where the DNA is. It's all this lacy chromatin stuff that you find out in the nucleoplasm. And that's where it's going to start condensing up and forming into structures that are easily identified, which are the chromosomes, okay? And they're going to move to opposite sides. And it says of the cytoplasm here because the nuclear membrane starts to break down at that point, okay? So it really starts in the nucleoplasm, but becomes the cytoplasm as the nuclear membrane disintegrates. Metaphase, we'll see now that the chromosomes are easily identifiable, they're going to be pulled to opposite poles by spindle fibers. And then in anaphase, we're going to see the chromosomes are totally separated and moved over to the poles, and the centrioles start to shorten up. And then in telophase, we're going to start having the new nuclear membrane forming around the two separate total genomes. All right. Interphase, that part that came before, if we go back here and look at this slide, interphase, this is usually the cell that you see under the microscope is almost always in interphase, okay? And that's where the cell is really active in starting to replicate the DNA and getting prepared for cell division. It's doing all of its business, its growth, its multiplication of the DNA, synthesizing all the new particles that it needs in the new cell, all right? So we divide interphase into these phases, all right? So the S phase, synthesis of DNA, all right? And then we'll go through these growth phases, sometimes called the gap phases. And this is where we prepare for all the changes that will undergo in mitosis. All right. And if you look back, you can see here, you have the G1 growth phase here. You have the S phase where we start replicating all the materials. G2 is right before we enter mitosis. All right. And this restriction checkpoint is often called G0 or G0, okay? And that's, we kind of leave the cell cycle for that checkpoint. All right, so kind of learn your phases right here, and we'll look at what they look like now under the microscope. So in late interphase, we've already passed the restriction checkpoint, and our DNA has checked out as it's okay. We don't have any crazy mutations that we need to take care of, all right? And so we're going to get ready for cell division. So you see the nuclear membrane here should have been disappearing. Our centrioles are starting to align to get over to the opposite sides of the cell. And then we're going to start pulling apart as we get into later prophase. Later interphase and enter prophase, we're starting to get our poles, disintegrating our nuclear membrane. We have strictly formed chromosomes instead of that lacy chromatin, all right? So now, you know, you have 46 chromosomes. Here we're just showing 4 of them. It's going to pull our chromosomes, start getting attached to the spindle, and we're going to start lining up down the center here in late prophase. Now we have our sister chromatids all lined up, okay, down what we call the metaphase plate. You can put like a whole plate separating the 2 chromosomes right here. This is your marker for metaphase, all right, when they all line up. And now anaphase is we start separating one sister chromatid from the other sister chromatid over here. And we're going to end up forming 2 completely new cells. All right. So anaphase, you can see it under the microscope here, anaphase. Here's the centrioles kind of pulling away. Here, they're still lined up on the metaphase plate. Telophase is marked by, you can see the difference from anaphase is they're really pulled to the opposite sides, and you can see cytokinesis happening as the cell starts pinching in and forming new membrane, okay, around it. Okay, then we're getting back after we have this cleavage furrow into 2 new cells, we're back at interphase. Now we're more at the G1 phase, right? Where cell starts conducting its business. It'll eventually start replicating the DNA and then get into the G2 and late interphase up there. All right. Cytokinesis is different from nuclear division. It just means the cytoplasm. It starts in anaphase. Where you really see it is in telophase, when you see it starting to pinch in this membrane here. You get this contractile ring forming in here. It's going to be made of like actin, actin filaments that are in your muscle cells. They're going to pinch in and pull that cytoplasm in half. And you're going to have the newly formed cells that are each going to be identical in their DNA. The cells are exactly the same, or they should be, because they've made it through the checkpoints at the cell regulatory control. All right, so this whole process of cell division is highly regulated, varies a little bit from cell type to cell type. You'll see Skin cells, intestinal cells, blood forming cells are constantly going division. They're constantly dividing and forming new cells. And think about it. Your skin cells are always sloughing off, right? They need constant replacement. Your intestinal cells are kind of like the internal skin cells. They're constantly sloughing and being replaced. This is also why when we do chemotherapy, what cells are affected most by our chemotherapy agents? Rapidly dividing cells, okay? This is why you get sick with chemotherapy. Your intestinal cells are rapidly dividing. Our chemotherapy agents go after rapidly dividing cells. So your intestinal cells are very much affected and you get sick, nauseated, and you vomit. Your skin and hair cells are a modified skin cell. These are also rapidly dividing, constantly growing cells. Our chemotherapy agents target rapidly dividing cells because then skin cells, what happens with chemotherapy? Your hair falls out, all right? Why chemotherapy targets rapidly dividing cells? Because cancer cells are rapidly dividing cells. So a lot of our chemotherapeutic agents go after targeted cell division, rapid dividing cells. Cells like neurons, you know, don't divide rapidly. And this is why we have a hard time when you damage your neurons. We can't really do much to help replace them or have good therapies for it. So you sever your spinal cord, you know, you end up paralyzed. We have a very limited ability, but getting better, using some stem cell research in that, stem cells divide rapidly, to start replacing some of these, okay? All right. Mature red blood cells don't have a nucleus, all right? These cells, when they're born out of your bone marrow and they come out, they'll have a nucleus, at which point we call them baby red blood cells or reticulocytes, and they rapidly lose their nucleus because they live for about 120 days, all right, in your blood. You're constantly replacing them. So the average pool life is somewhere between 90 to 120 days on your red blood cells. They lose their nucleus because they become a streamlined organism to be full of hemoglobin that carries oxygen to all your tissues. And that's pretty much what a red blood cell does, all right? So they can't divide or replicate. So when you lose your red blood cells, this is why we have to give you red blood cells from a donor, okay? Your body will start, you know, usually making new red blood cells, but it takes a little bit of time, okay? So that's why we give them from a donor. All right. When we start looking at our dividing red blood cells, we'll see that at the end of the chromosomes, we have a region called the telomere. It's like a little cap. And with every cell division, you kind of progressively lose a little bit more of your telomeres. This is a process of aging. So I probably have short telomeres on a lot of my cells compared to most, I think everybody in the class, unfortunately. Your telomeres shorten with age, all right? And this is kind of like senescence as you move towards aging and death. You have less of an ability to replicate and divide your cells, all right? So progressive shortening of telomeres leads to senescence, apoptosis, planned cell death, we'll talk about that, and oncogenic transformation. We start finding less fidelity in that replication process, and we might skip over that checkpoint, and cancers tend to flourish a little bit more, show up a little bit more as we age because our cell division control is not as good. All right. So throughout cell division and the control of cell division, we're going to have different proteins that come into play at different stages. And our cells divide, they grow, they get bigger, and then they need to divide because they don't want to keep getting bigger and bigger and bigger. They need their favorable surface area to how much volume. They want to maintain their size and shape. All right. And it's going to be a lot of hormones and growth factors that help give signals for this control on cellular growth. The other thing that helps with cellular growth is this concept of contact inhibition. Cells kind of are inhibited in their growth by everything that kind of is in their neighborhood. It limits how big the cell gets, the kind of touch points with other cells. And even in a lab, when we put cells to grow on a culture plate, you put a couple of cells on here, and then they'll grow and they'll grow, and they'll grow in a monolayer across a plate until they reach the edge of the plate, and then they stop. Cancer cells have lost contact inhibition, okay? Cancer. And they will grow and grow and grow across the plate until they actually mound up, okay? And they'll grow over the plate, and they grow not in a monolayer. They grow all over the place. Normal grows in a monolayer, okay? Has contact inhibition. This is because tumors have that loss of control over their mitosis. So that's one of the ways we can see cancer cells. And those of you who've seen the things about Harriet Lacks and the HeLa cells, these were a cervical cancer cell, and they saw that they lost this contact inhibition. They kept growing. We call it immortality of cancer cells. Okay, they just keep growing. They don't have the normal stops on that. And that's what makes those cells useful in a lab situation, that we can keep watching them grow and grow and divide, and we can manipulate the DNA and do a bunch of research and stuff in immortalized cancer cells. Let alone, sometime we'll talk about the lack of ethics in not telling Miss Lax that they were using her cells for these purposes. Hopefully we're getting better at some of the ethical issues, but oh my God, let's take a little aside on ethical issues. Did any of you read or hear the thing in the news about these two nurses in Long Island that started this scam that began right around Thanksgiving time of fake COVID tests? And they were having a whole bunch of people come to get not fake COVID tests, fake vaccine records. So they created little fake vaccine cards, or maybe they had real vaccine cards that they then sold to people who didn't want to get vaccinated, and gave them vaccine cards so that they could go to restaurants and, all the places that you needed to produce a vaccine card in New York City or the greater New York metropolitan area. They made $1.5 million from Thanksgiving into January when this was discovered. I was like, holy moly, that's a lot of vaccine cards. That's a lot of effort for people who just didn't want to get vaccinated. And I was so disappointed because I think I told you before, I'll criticize doctors all the time. I usually won't ever criticize a nurse, but this case, I'm criticizing the nurses. It's like, I used to do a bunch of forensic work. And one of the things is, a nurse coming into court is like the most believable witness on the planet. Juries believe a nurse way before they'll believe a doctor or a dentist. They kind of think that there's too much money involved in being an MD or DDS, that, that kind of corrupts people and makes them try to hide things. They will almost always believe a nurse. But the more we see nurses doing crap like this and getting out there, we're going to also start finding nurses to be less credible to juries. So police your own, you guys, when you get in there. Police your own so that you still bear the honor in your profession. I get sad when I hear all the stuff like, we had years ago, I forgot what his name is now, but the cancer doctor here at Carmanos in Detroit, who was giving people chemotherapy when they didn't need it. And then the ones that were supposed to need chemotherapy, sometimes he didn't have all the agents and gave them saline solution. So people with unnecessary chemo, people not getting their chemo, but he's getting rich. It's disgusting. We have to keep the honor in our profession. The public expects it from us. So terrible stuff.
It's like,
right.
why? Oh, I'm sorry.
Go on.
I was about to say, like, why risk it like so much? Like, why? Like, what?
I don't know. I personally,
All
I just
for $1.5 million.
Yeah, 1.5
Yeah,
million.
exactly.
But it's also like, how do you live with yourself?
Exactly.
This is like people's lives. These are, don't they have people in their life that they care about? Like, well, how would you feel if this happened to your grandmother or your baby or something like that?
Exactly.
You always have to, that's the one thing about healthcare is we're supposed to be an empathetic profession. You're supposed to be able to put yourself as if you were in your patient's shoes
Exactly.
and, treat others like how you would want to be treated.
That's why this profession is not always for everybody, because your empathy is what stops you from letting your stress affect your bedside manner.
Right.
And so
and sometimes it will, and sometimes you apologize. You say, I'm sorry, last time I saw you, I was a little short with you. I had 27 patients I had to see that day, and I just didn't have time to listen to everything you needed to say, but I have time right now. And through COVID and how understaffed everything is, we do a lot of apologizing now to patients. You just, you have to serve everybody and you have limited resources to do it. And by and large, people understand that, the public doesn't seem to understand it enough with the stress that healthcare workers are going through. We've had doctors and nurses committing suicide early on in this because of the stress and you get used to seeing patient death in the hospital. You even get used to, like when I worked in the ICU, you get used to seeing it every day, but it's usually like one patient a day. It's not 20 patients a day. That I don't think you ever get used to. So that's a really, really difficult thing. And it's hard because, we get used to feeling like, sometimes people come in and, there's not much you can do to help them. But most people we can help. But early on in COVID, we didn't know what to do. We didn't know how to treat them. And so it was like, very overwhelming to feel like, here I am in this thing and I can't stem the tide of patients coming in and I can't really help the ones that are here. And it's very frustrating for people who are used to trying to solve these problems. So it's there's so much that's wrong with our healthcare system that I hope all of you can go on to helping to fix it. And taking the money incentive out of healthcare is going to be one of the big things. And making sure also that people are well remunerated for their efforts. There's no reason why a neurosurgeon should make $500,000 a year, but your pediatrician who works just as hard makes $75,000, $80,000, $90,000 a year.
Yeah,
it's
I agree with that. It's
crazy.
Kinda...
So, or your LPN is making 40 grand a year. There's no reason that they are very underpaid for what they do.
I heard LPNs are being phased out now.
That's the rumor. They're being phased back in. There's a bunch of new
They're
LPN.
being used. At least St. Joe's is starting to utilize them to fill in the gap of labor shortage for nurses.
Exactly.
So it's a
exactly.
limited role.
Well, they are nurses. They're
They
not,
are.
they are nurses. They're licensed practical nurses. So they're usually like a year of training instead of like 2.5 years to get to be an RN.
What's the difference between that and CNAs?
So CNAs really is more like on the job training, and then they take a test, and their test is very limited. They don't have to know all the like chemistry and you know, I have a lot of CNAs who come back for training to either be an LPN or RN. CNAs is more practical of assisting a nurse to do like a lot of basically the grunt work, the scut work of nursing, the changing of the beds, the changing of the diapers, the, you know, more simple things that don't require a lot of thinking. And I don't mean that to insult that the CNAs can't think. But you get paid progressively more and you have higher licensure, the more your job requires you to use this, okay? So an LPN can pretty often do some things like at some places not, starting an IV, you know, some places only allow an RN to start an IV, and then you get an RN who can do a whole lot more, can make some decisions like, you know, the doctors can leave orders and the RNs can decide what medicines to dispense and when to dispense them under cases of like, you know, pain, constipation and all that stuff at the bedside. They make a lot of decisions about patient care that are then implemented by the LPNs and the CNAs. And then you get your advanced practice nurses that, you know, basically function almost as a quasi doctor or physician extender. So it gets into a lot, you know, a lot more and more stuff. You know, a CRNA in the OR completely administers anesthesia and makes all the decisions about the medications to be administered. There's usually a circulating anesthesiologist who goes around and supervises, but, you know, good long-term CRNAs are working pretty independently, just like a nurse practitioner in, you know, your local family physician's practice. You know, they carry their own patient load. They make their own decisions about the patients. If they have something that's really complicated and out of the domain of their knowledge, then they go and they confer with the physician. But often it's just a conference and they carry out the plan that the two develop together. So they're getting more and more ability to practice independently. All right, so let's get back to ourselves. All right, so sometimes we get the cancers, right? The cancers are cell growth gone awry. None of our cell control points remain, all right? So sometimes these are benign, which means the tumor tends to remain localized, all right? But sometimes the cells kind of split off and travel through the blood. They travel through the lymphatic system, or they can even invade locally through what we call, when we get into the tissues, the basement membranes that separate different tissue types. And that is malignancy, all right? Now, sometimes we have in the DNA underlying things that cause the tumors to grow and flourish and spread. So the difference between oncogenes and tumor suppressor genes. Oncogenes are when we have our genetic makeup kind of changes and you have a mutation, and suddenly you might start expressing, say, a protein at a really high level, and that can be toxic to the cell. It can transform the cell into being a cancerous cell. It can be something that helps the cell evade all those normal checkpoints on the cell growth, all right? And you see that here. Here's like some normal cells with all their hair-like little microvilli. This, to me, these look like respiratory cells or something. And then you have this tumor growing in here, and it doesn't have the normal projections of the cilia or microvilli, whichever it is, on top of these cells. All right, so you can even see that they look different. That would certainly affect the function. Oh, it says it's cilia. Okay, so cilia in the respiratory system beats up all the mucus and bacteria so you can expectorate it. All right, if you don't have that, you have dysfunctional cells in your respiratory tract. They aren't going to have the cilia on them to beat up and move out the viruses, the bacteria, and allow you to, cough it up, expectorate it from your system. Tumor suppressor genes are the normal genes that we have, okay? Those are the checkpoints. This is what limits mitosis. This is what tells our cells, stop, there's something abnormal going on. But when our tumor suppressor genes get a mutation that makes them not suppress, makes them not go through the checkpoint, then we end up with uncontrolled mitosis that way, okay? So your tumor suppressor gene can almost become an oncogene by not working the way it's supposed to in suppressing mitosis. All right, so the idea of a cancer trigger, we call this often the two-hit theory of cancer. All right, so usually it's the idea that you're born with one gene that's bad, one bad gene. And then somehow in life, you encounter something like ionizing radiation that they're showing here, or maybe you encounter a toxic chemical, and so then your second gene goes bad in life. Second gene goes bad. All right. Now, if you recall, you always have, and I know this writing is terrible this way, when you have your two genes, like let's say you have chromosome 10, right? And if chromosome 10, you get two genes, right? One from mom and one from dad, right? So let's say you're born with the bad gene from dad, I should say, because, we always blame the mother, right? It's very Freudian. So if you get your bad gene born here from dad and it's bad, but you still have a good gene from mom, usually that's enough to make the correct proteins and stuff to get you through life okay. But then if mom's gene gets ionizing radiation or something and the gene goes bad, then you have two bad genes. Suddenly you're not making any of the right proteins. Everything goes haywire, okay? All right, so this is the idea. You can have, and they can be different genes. Like if in mom's, like if you were born with an oncogene on dad, on your chromosome 10, and then if mom, the tumor suppressor gene goes bad, that too, both take out maybe the genes that are vital on, say, chromosome 10. All right, so here's your two-hit theory. Oncogene is turned on. All right, tumor suppressor gene gets turned off. Suddenly then your cell goes through uncontrolled mitosis, and it's spinning around, dividing really, really, really fast. It's not making all of its vital proteins, all that, and your cells become abnormal. Instead of being, shaped and having all the functional stuff, maybe having all the cilia that it needs, whatever it's specialized to do, these cells become unspecialized, undifferentiated, lack the normal proteins. And sometimes they split apart from each other and they start to travel through your blood system and they spread. They spread all around your body that way. And that's how you end up with what we call distant metastases. It started as cancer in your liver, but now it's spread to your bone and your brains. All right. This is kind of cancer in a nutshell here. Okay, so I just mentioned stem cells. So stem and progenitor cells. We start out with cells like our early egg and our sperm meet and form our zygote, right? Which goes on to be the fetus, and suddenly the fetus differentiates into all different types of cells. We start differentiating into nerve cells, bone cells, digestive cells, respiratory cells. That's the process of specialization. But early on, the cells could become anything, okay? That's the idea of the stem cell, the cells that can flourish and become multiple different tissues, okay? So the options of a stem cell is they can divide and form 2 new stem cells. This is that self-renewal, so that you get a pool of stem cells, all right? They can also go on to form what's called a progenitor cell. This is kind of the first stage in differentiation. This is a cell that's going to go down like a specific line to become a specific type of tissue, okay? Like maybe it'll go on to become skin cells or something like that, all right? So it's an early progenitor, early stage of differentiation into skin cells. All right, progenitor cell, specialized stem cell. It's still a type of stem cell. It's going to go on, like I said, to become committed to the skin line. All right, but it early on can split into being skin cells that are totipotent. Okay, totipotent can become sort of like any kind of early cell, or pluripotent, limited to specific cell lines. All right. All right. So stem cells of later development. These are the progenitor, and I should have put this here first. Totipotent and pluripotent. Pluripotent maybe decides to go down the skin line. Totipotent can become everything else. All right. As we get farther in the differentiation process. All right. So here we see the stem cell going to the pool of many other stem cells that can be anything. Stem cell goes on to become the progenitor cell. This progenitor cell can be either totipotent or pluripotent, okay, depending on its level of differentiation. Totipotent kind of can go on to become a lot of different things, Pluripotent becomes a little bit more specialized. In this case, it's going to become white blood cells. All right. All right. So now as it differentiates, it's going to mature in specialization. Putting it together, like I said, we have sperm cell meets egg cell. We have our early fertilized egg. All right. From the early fertilized egg, if we separate the first division, okay, the first division into 2 cells of fertilized eggs, when it divides, if we separated these 2 from each other, this one can become a whole human being, and this one can become a whole human being, and that's how you get an identical twin, okay? That can happen early on in the pregnancy, okay? That's why they're identical. They have the exact same DNA. It happened at an early cell division. Later on, we get to stem cells that are self-renewing that get down to this progenitor stage. It's not going to be able to become a whole human being, okay? So we have the progenitor cells that go down the line of becoming skin cells. We have the progenitor cells that go down a separate line of becoming the nerve cells. We have those that come down kind of in that same line a little bit, it's nervous tissue early on, but this one goes on to become what we call the supportive nerve cells, our glial cells, and what become the actual functional nervous system cells. Regenerative medicine is the area of medicine where we're studying all this stem cell technology. This gets into a whole lot of ethical discussion, and people fall down on different lines of where you feel this is okay. All right. And I'm not even sure where I feel on all of this at different levels. I change my mind all the time as I read different things. So you can get donor stem cells from things like the umbilical cords. These tend to be not quite totipotent, but a little bit more pluripotent kind of cells. And we're using a lot of these after, they'll ask you if you want to donate your umbilical cord to stem cell research when you have a baby. And so a lot of people do these. A lot of people bank these in case there's any problem in their own families now that they can get some donor stem cells. We can also get stem cells from a patient from natural sites in the body where the stem cells exist, such as in your bone marrow, and we can try to reprogram them to become a different type of cells instead of the cells that are normally differentiated from bone marrow, like your red blood cells, your white blood cells. The other, I should put down here, sources of some stem cell is, of course, fetal cell research, okay? The book didn't put it in here because it probably didn't want to get into all of the ethical complications with this. And this would be fetal cells that come from natural miscarriages, okay? Miscarriages or from aborted fetal tissue, all right? And some women kind of feel that when they've had an abortion, one of the things that they can do is donate the early tissue to stem cell research. And of course, there is a big brouhaha over that, as especially now the whole ethical and legal quandary over abortion is right before us as we have a whole new conservative Supreme Court. I can tell you that you can look it up in New Mexico, where I used to live. The University of New Mexico got into trouble with the local population over getting fetal tissue donated to some of its research projects from local abortion clinics. It's a highly Catholic area of the country, so that was very problematic for the university. All right, cell death. So apoptosis is what we call programmed cell death. This is the process the cell undergoes when it's, it kind of surveils in the homeostatic system for cells that aren't doing so well, cells that might have accumulated a problem in it over time. It's also like how we differentiate for the developing fetus has programmed death in specific areas, such as, removing the webbing between toes as the skin is growing in the fetus. We program it so that we have the body outlining contours and the cells are removed that are growing in places that we don't want the cells to be growing, okay? We have programmed cell death after you get a sunburn and you want to peel away those top damaged layers of skin. So this is like the homeostatic mechanisms, as I said, that just control and check for abnormalities, cells where we don't want them. We get rid of the cell in a specific controlled manner, all right? Necrosis is when the cell gets so damaged that it just has to die. And this can be, damage from something like a massive burn or toxic chemical. It can be from microbial infection. We try to control in some ways. If you get into microbiology, you'll see that we release things to get an apoptosis in some cells that are infected, so we can control the death of that cell and not have an infection flourish. But sometimes you just get kind of a massive death of cell that is in an uncontrolled fashion, and that's necrosis. All right, so This is some slides they put at the end here of stuff we've gone over, and then we have some practice questions. But we're not going to do that right now because we just have limited time. We're going to spend the next like 15 minutes or so going over the remainder of chapter 4. Okay. So let me get up chapter 4 where we ended. I think I put an endpoint on here last time. I thought I wrote in on the slides. Actually, where we stopped, it was enzymes. Okay, let me bring this up. All right, so we started on enzymes, and we talked a little bit about how enzymes are protein catalysts, how they lower the energy of activation for a reaction to make it more likely to go over. That was back here, this whole bulldozer idea that normally a reaction wouldn't take place because it has to overcome this big giant energy hump, but the enzyme comes in and it lowers that energy of activation, kind of bulldozes it down so that we have a much lower energy barrier to overcome with the enzyme so that the reaction is more likely to take place and form stable products. Energy of activation is oftentimes written as EA, and that would be this whole thing right here, that whole energy of activation, and it gets lowered. All right, so how enzymes work as protein catalysts, they are a protein. And what they're going to do is they're going to take the substrate molecule, the molecule that we're trying to change into something else, and it's going to usually hold these substrate molecules close together. That's what lowers the energy activation. It kind of tends to bring those molecules together so that they're more likely to react with each other. And it's called a lock and key hypothesis, that these things fit into these little pockets on the enzyme in just its sole particular manner, okay? And then it's acted upon to change it into the product, which then the enzyme can have the product drop off of it, and you have the enzyme left at the end of the reaction, unaltered and ready to act with another round of substrates. All right, things that can affect how the enzyme acts. Too much heat. Too much heat can denature the protein of the enzyme, change its shape so that it no longer fits in that lock and key. Radiation can basically do the same, alter the proteins. Electricity can alter the way the actual chemicals interact with each other, same as with specific chemicals. Or pH, pH can change, the way like the hydrogen bonds might form. Poison the protein. All these lead to, can be like excess heat, radiation, leading to denaturation, changing the protein, altering it somehow so that we don't have that fine lock and key fitting. And we discuss, like egg albumin, when you heat it, you denature it, you change the protein, it becomes solidified white. You can never change it back. Sometimes you can renaturate, sometimes you can't, depending on what happened to the protein. The human metabolome is kind of like the concept of the human microbiome. The microbiome is all the microbes that live in us pretty much harmoniously for the most part. The metabolome is all those small molecules that are part of our metabolism that exist in the cells, the tissues, and the larger organs and organisms. We're starting this human metabolome database so that we can look at all these chemicals or metabolites that we have. It's estimated that there's about 2,500 of them, okay? And so we record more and more about what are the normal chemicals involved in the normal pathways so that we can also utilize to see what happens in the pathological situations, all right? Not the crux of our information here. All right, so we need to understand metabolic pathways and how they're controlled by enzymes. Okay, so enzyme controlled. And that's going to be that some specific metabolite is going to act as a substrate. And it gets acted on by an enzyme, which turns it into another product, which can be the substrate for the next enzyme, okay? Which gets turned into a product that becomes the substrate for the next enzyme. And there can be a whole chain of these reactions till we get to maybe a final end product in a series of metabolic pathways, all right? So it's usually, we have long metabolic pathways to get to any endpoint in our body. And of course, things can go wrong at any level here of any enzyme. If you take out enzyme C, for instance, we will get enzyme A and enzyme B acting till we get a buildup of substrate C, but we can't convert it into substrate 4 or this final end product. Sometimes substrate C3 will build up and will turn off this enzyme or this enzyme, okay? Because we don't want to keep building up this thing that we've already got a lot of, okay? And that is called negative feedback, right? Negative feedback, where something later on turns off the earlier stages here, okay? So we often put like a little negative sign here. All right, all these enzymes that turn substrate 1, substrate 2, substrate 3 into their products typically end in ACE, okay? That's one of the ways you can tell that it's an enzyme. Lactase, okay, protease, lipase, lipase acts on lipids, protease acts on proteins. Lactase specifically acts on the sugar lactose, maybe. All right, so every step is controlled by a different enzyme, okay? And you have a whole pathway. And as I said, the product can feed back negatively. That's a negative feedback loop back on, the initial enzyme. It can also feed back on later enzymes, okay, as these products build up. And in some pathways, when the product feeds back on maybe the first one, maybe this would be the rate limiting enzyme. It's the enzyme that tends to be the control point in the whole sequence of reactions. So don't always think that it's necessarily the first enzyme. Some instances it might be this enzyme or the enzyme right before the product, but it's what's kind of ends up being the control point for the whole series of the reactions. It's usually the one that has kind of the most difficulty turning into its product, all right? The rate limiting enzyme. All right, so we have our metabolic pathways here. Here we again have enzyme A being the rate limiting enzyme, and how you see it might work in a biological system is we have substrate 1 out here in the extracellular fluid, and it gets acted on still outside of the cell at the cellular membrane by an enzyme protein that's an integral, membrane protein right out here, the first step. It kind of spits it over to substrate 2 that'll get acted on by another enzyme, acted on by enzyme C for the third substrate, acted on enzyme D, which can then form a product that maybe is diffusible to come through to the intracellular side. Here on the intracellular side, it might actually inhibit the action of enzyme A. Now, it doesn't always have to act in this way. It can be, like I said, many different ways, but something is going to limit, whether all of this pathway continues to work. You will need to be told that. There's no way that you're going to know which one is the actual rate limiting step at your stage. Oftentimes, the enzymes in trying to convert one substance to another, along with the enzyme, they'll need cofactors, okay? Cofactors are substances that interact with that enzyme to help make it active and act on the substrate. Sometimes it helps the enzyme kind of have a little bit different shape for that, make that lock and key kind of conformation. Sometimes it helps to bind the substrate in a little better, and it can be anything. It can be an element, it can be an ion, anything can kind of be a cofactor. We call it a coenzyme when it's an organic molecule that acts as a cofactor. These are things that we typically think of as like the vitamins that we take. Vitamins are often like, essential cofactors in a metabolic reaction that help that reaction take place. This is why people often take vitamins thinking it's going to help with their metabolic processes to work a little bit better, okay? And of course, denaturation, like I said, can happen at any point along the way. Is that this is what happens to inactivate the enzyme. So you can have a poison come in that might even sit on there and look like a vitamin, but it's a little bit tweaked, and it actually inactivates the enzyme, changes the shape of the protein, makes the enzyme unable to bind to its substrate. Okay, so there's that picture again, just to remind us. All right. When you are born with a defective enzyme that doesn't work right, and it can't change your substrate into the product that your body needs to use, we call these inborn errors of metabolism. All right. So this ends up resulting in the accumulation of the substrate, because if you have defective enzyme B, like I said, substrate 2 will build up because you can't turn it into substrate 3. PKU, phenylketonuria, is an example of this. This is something where people are born with a defective enzyme that turns the phenylalanine into tyrosine. You don't have that. So you don't go from phe being the substrate to tyr. That enzyme is defective, okay? And so you get a buildup in phenylalanine, all right? Excess phenylalanine builds up when it's at a high level, is toxic, and it will poison the brain, sitting on the brain cells. So how we test every baby at birth for this, because we can, and we know then immediately we need to put them on a special diet that's low in the amino acid phenylalanine, okay? All right. Energy for metabolic reactions. So energy is the capacity to change something or do work. That's always the definition you learn. But this always seemed to me when I was a beginning student like a nebulous definition. This didn't help me much. And we know energy comes in different forms. We can have energy that's heat, right? We have energy that's, fossil fuels, gas. We can have energy that comes in that's electric. We can use sound, harness sound as energy. And energy is this concept of it supplies us with that, ultimately the energy of those kilojoules in our bonds between our chemicals, and it cannot be created or destroyed. We can just change it from one form of energy to another. So we can take heat energy and harness it and utilize it, and we can turn it into electric energy in our house to drive all of our electric products. That's what we do when you use solar panels, and we convert it and use it to drive our, electricity in our house, for example. All right, so energy, as you know, is basically in the form of ATP in our cells, and we will use it to drive our chemical reactions, all right, to make those take place. When we're talking about using ATP, pretty often we're talking about having it drive the reactions of cellular respiration. And this is where we're going to transfer energy from all those bonds that are held in our molecules and make it available for cellular use. And usually we're taking the energy off of that high phosphate bond at the end of ATP, capturing and harnessing all the energy that's held in that last bond. The ATP becomes ADP, and it splits off into that inorganic phosphate, but all the energy that was in the bond that held that phosphate to the ADP, making it ATP, drives the reactions to go forward in our body. And I know this is kind of nebulous, kind of a hard concept to grasp. All right, so our chemical energy stored in ATP, stored in that last high energy phosphate bond that we put like this, that when we break it, makes ADP, right? Okay. Oxidation is the process that we use to release energy from the food, like the glucose and other molecules that we eat, right? We take in our glucose and we're going to break the bonds in that glucose. And we're going to use that energy also to drive a lot of our reactions. That's our cellular metabolism, okay? In cells, our enzymes are going to lower that activation energy in order to drive these oxidation reactions. It breaks apart all the energy held in those glucose bonds. And we call that cellular respiration, okay? Breaking apart our glucose, we're going to trap all the energy that was in the bonds and all that glucose. And we're going to want to transfer it to the pool of ADP that we made from all our other reactions that used ADP. And we're going to try to build that back up to harnessing all that energy from the glucose to reattach some of this phosphate back to ADP to make it into ATP, okay? So this is how we're going to energy being transferred. I shouldn't say to ATP, but into building ATP. Energy is transferred into building ATP, all right? From glucose into making that bond. In ATP, 40% is going to be released in that chemical energy that we're going to harness in the bonds that are going to build the ATP. But as we go through all these reactions, you know, extracting all this energy from the glucose, some of this is going to be dissipated as heat in our body. In fact, more of it, 60%. But heat in our body is important. This is why we are warm-blooded animals, right? Okay. All right, so we looked at ATP a little bit last time. Let's remind ourselves constantly, okay? ATP carries cell energy in the form that we can use. That's why we call it the currency. We're always taking off the PI, putting it back on. And this is why we call it currency, is like we either deposit money in the bank, putting PI, the inorganic phosphate, back onto the ADP and making it into ATP to store for later use, or we're making a withdrawal. We're needing our money. We're needing our energy right now to drive a reaction, and we're stripping off a phosphate right here, okay? And rendering our ATP back to ADP and inorganic phosphate. And we're always going to be extracting the energy from that bond right there. Yesterday, when I do the same thing here at the beginning at both micro and at anatomy, a little bit different focus on each one, but adenosine, someone was asking me, what's the difference of adenine and adenosine? This is your nucleoside, okay? Nucleoside is when you have the nucleotide attached to the sugar. In this case, it's ribose. Adenine and ribose. You could have thymine and ribose, right? Okay, cytosine, guanine. Your nucleotides are adenine, guanine, cytosine, thymine, uracil, right? Okay, so you have it attached to ribose in the case of ATP. When we have it, we also have it in our DNA, right? We have adenine attached to deoxyribose. That's the nucleoside, okay? All right, and then we have in ATP 3 phosphates on the chain, all right? So it's usually the second and third phosphates are attached by these high energy bonds, the squiggly line. This one being higher than this one. Here's the third phosphate, second phosphate, first phosphate. If you just had this AMP, this one ADP, all 3 ATP. We call this ATP ADP cycling. We're going to end up going till noon today, all right? But you don't have lab. All right, so when ATP loses this terminal phosphate on it, it becomes ADP. It can, of course, get converted back to ATP by adding the inorganic phosphate that we abbreviate PI a lot. All right, so this is your cycling. ATP, we're going to expend it. We're going to get rid of this PI. We split it off. This energy gets utilized to do other reactions. Now we're here back at ADP, right? 2 phosphates. When we eat food, we want to harness the energy from glucose or something like that, our C6H12O6. We go through the cell cycle, breaking down this glucose, and we make some ATP, we make some inorganic phosphate here, and we'll attach it back on once we get over to the electron transport chain, for instance, to go back and make ATP, all right? So when we break down glucose, we're going to build up ATP. Other cell reactions will spend ATP using the energy in this phosphate to break it down. Sometimes even in the process of breaking down our glucose, you'll see, we'll have to use some inorganic phosphate from our ATP to break down some of these glucose bonds, but ultimately we will make more ATP than what we use in breaking down the glucose. That's how we extract some energy, if that makes sense. The idea that sometimes you have to spend some money to make some money, like you have to buy a car so you can go to work to make more money. That's the concept here in ATP.
Do we still get energy from ADP, or once we break one of those phosphates bonds and we go down to ADP, there's no more energy to be had from the...
Good question. When you break this last bond, you get many, many more joules of energy. So we prefer to break down ATP into ADP. Sometimes we also go as far as breaking down this third bond down to AMP, okay? And then we have to phosphorylate it twice and have to use a lot of energy to recover the pool of now AMP to build it back up into ATP.
So
but
it's a lot less efficient.
It's a lot less efficient, exactly.
Okay.
So this is kind of like your old clunker car. This would be like your brand new car that you had to pay a lot of money for on a lease. It's going to function a lot better, all right? So, all right, so ATP is like, in this example, like a charged battery, fully charged. You know, you've left that cell phone on your jack all night long and it's full charged up. You can use all the energy that's in it. ATP's not quite like a dead battery, but like an almost dead battery. So you can extract a little energy off of it. But once you take that energy off, that cell phone, you know, you can make that last final call. Okay. Think of it more that way. ATP is hydrolyzed. We break that bond, right? Use water, break that bond to split off that phosphate and capture the energy. In ADP, once again, we can recharge it by adding the phosphate back to it. We condense it with water to put that phosphate back on. All right. So we're going to release chemical energy from ATP that's held in the bond. We're going to break that bond, release the energy, and then when we need to recharge it, we're going to use the oxidation, releasing the energy from glucose, okay, to add back in energy from ATP. The energy is used to power all the cellular metabolism, all the reactions, anabolism, catabolism. We're going to have to burn some chemical energy in order to power it. All right. All right. The other player in all this are going to be some of our coenzymes. And the really important coenzyme is NAD and NADH. And this is, I'm going to try to get through this a little bit before we stop. And we'll come back to it next time when we have some questions, because this is always where people really don't understand this. When we get into the Krebs cycle in TCA, citric acid cycle, the carboxylic acid cycle, Krebs cycle, it's all the same thing, whichever you call it. The really important thing to pay attention to is the NADH and NAD+, the coenzymes. This is the oxidized form, and this is what's called the reduced form, okay? The reduced form. You've reduced it by adding hydrogen and the electrons from it onto this oxidized form that's deficient in electrons. Remember, it's got a positive charge, so it's deficient in electrons, all right? These are called electron carriers, this coenzyme, and it's going to be with trapping the energy from all these electrons that we ultimately will extract the energy from all these electrons that are being carried on these coenzymes. We'll transport that into our electron transport chain, which is where we make the ATP, all right? So NAD is floating around in its oxidized form, and it's going to trap the enzymes that we're going to strip off of, like molecules like the glucose, and we're going to trap it in this NAD now as the reduced form of NADH. This NADH will go into our mitochondria and donate the electrons to the electron transport chain where we make ATP. The other coenzyme carrier that appears is this FADH2. It does the same thing, but this is kind of the more common one. So you just think of this as kind of like a fancier NADH, and it actually makes a little bit more energy per molecule than NADH2, because you can see it's adding on 2 hydrogens. All right. All right, so we take those coenzymes after this is going to happen a lot through our glycolysis and our Krebs cycle. And we take all those enzymes that are attached to NAD, electrons that are attached to NAD as NADH or as FADH2, and we're going to pass them down the membrane, kind of like hot potato, from one enzyme to another. It's going to like extract all these electrons off in this process eventually that we call chemiosmosis, that will take the energy stored in this kind of proton, the hydrogen H+ gradients, as we strip the electrons off of it to make ATP. It's always explained so poorly. I hope I can do a better job to get you to understand this. All right. So you're going to see this whole cyclic thing, like where we take our pyruvate and we'll add some compounds to it. We'll come in here, we'll have like oxaloacetate, and we'll act on a whole bunch of enzymes in our Krebs cycle or TCA, acetate, the TCA, if you want to write this here, TCA. You'll see all these enzymes, but at different points, you'll see that we'll make NAD into NADH2, and it's just kind of thrown, sorry, as NADH. I'm jumping ahead of myself, and sometimes as FADH2, Okay, and those are kind of shown as these arrows, almost like they're a throwaway, but they're not a throwaway. They're the whole point of the cycle. These are the things that are then going to go on to electron transport. Okay, electron transport change, ETC. All this other stuff, these substrates, you can memorize them, and you know what? Your brain will evacuate them and you're going to forget them. I want you to really pay attention to where you're stripping off electrons, okay? Because that's what the cell utilizes. All right, so we're going to go through cellular respiration, and we'll go through the sequences of glycolysis, the citric acid cycle, and electron transport chain. We're going to do that the next time we meet because we don't have enough time to do it now. We're at noon. If you want to, you can look on YouTube, and I have from when I teach basic biology and I teach the electron transport chain, I have all these videos that go through it in a really detailed manner. And they're right here. You can go through the mitosis stuff if you want. You can go through the cell metabolism stuff. They're up there. It kind of really goes through it and explains it. I hope it explains it well, but you know, some things work for some people, some things work for other people. We'll try to understand it a little bit, and then we'll go through it next Tuesday and try to get to really understanding this. This is really important for like those of you who want to go into being like a nutritionist. I know I have a couple of people who want to do that. Anybody who's interested in like genetic and metabolic disorders, finally getting an idea where you're not really, I don't really want you memorizing all the steps of glycolysis and citric acid cycle, but where we get to looking at how those electrons come off and where those electrons will ultimately go. That's what we want to understand. That's like the knowledge that will stay with you over time. And that's what I'm interested in, is in long-term understanding, not memorizing when pyruvate becomes acetyl-CoA, becomes oxaloacetate, you know, and all those other chemicals. All right. All right. So we went a little bit long today. Those of you who have lab today, I'm not supposed to really be meeting and teaching today for things that are supposed to meet in person. I suppose if you have questions, you can contact me and we can check in, like maybe I'll get on Zoom at 3 o'clock. I'll send you all a link. And if you have questions about the lab, I mean, any lab that anybody's in, we can go over and answer questions at about 3 o'clock, but use your time to try to go through and do the lab. I have videos from the labs of the past when we just did the bone labs online. And so let me just show you where that is. In case you've forgotten, I know sometimes there's so much stuff on this website now, and it's really hard to find where everything is. Okay, so in the lab folders, when you click on over labs for mixed mode, if you go down here and we're in lab 3 and lab 4, axial skeleton, the things you can do to prepare for answering all those lab questions and learning the bones. When you go into what you're working on is this Grossman lab manual. In here, this is me in 2020 going over the axial skeleton with a group of my students. Now in lab, we go over these same things. I pass out a handout for what are the bone markings. You have that here on the website too, the list of the common bone markings. In the pre-lab slides, here's the slides for going over the axial skeleton and also the skull separated from it, which is part of the main axis axial skeleton. And here's the common bone markings handout. Okay, get a handle on the axial skeleton, and then you come down into lab 4. Let's go back one more here. Lab 4, the appendicular skeleton, and same thing, okay? you have your appendicular skeleton from Grossman's lab manual. And we're going to have the video that goes over all the appendicular skeleton. Those who were in class last Thursday, we didn't quite get to it. We took a little bit, maybe too much time doing our quiz or something. Tuesdays, we got through it. So here is the appendicular skeleton PowerPoints. So go through that, go along with the video, hopefully explains how the bones fit together and all that. I'll bring up my bone box. I have some bones here at home, and I'll bring those up with me at 3 o'clock. And if you want to meet and we want to kind of try to fit things together, we can do that. LaShonda is entering. Did somebody ask a question?
Does our PBL start tomorrow?
No, I said at the beginning of lecture that I'm going to, and I'll put it in the announcements, that we're going to hold off and do the PBL next week. We're a little bit behind on time with the, I tried to figure out where things were going to land in this 14-week semester versus the 15 weeks, but I didn't quite estimate right, and I didn't account for a snow day either. So we'll meet next Tuesday, and I'll plan on opening up the PBL on Tuesday night, and you can start working on the questions then, okay?
Okay.
And I'll give you still like 5 days to do it. All right.
Okay.
All right.
Okay.
So here's where the appendicular skeleton lab that you have to hand in is. And I think you can go from there. And like I said, especially for those of you who got snow day today, going through some of those videos, if you can go through them, I'm trying to find them up here. I think I put it back. Go back. Bone videos like this on appendicular skeleton. If you go in here to like lower extremity, this will play like the femur, and it goes through and shows you all the parts of the femur, kind of like what you would do in lab. Now, a lot of these are MPG videos, so you might have to translate it into MP4 if you only play MP4 on your computers. It wouldn't come up in Firefox in the lab videos. And I suppose if we get into real problems with that, I can try to translate them and post them on YouTube or something like that. Okay, but you can see it's really helpful going through all the parts of the bones, pointing out the bumps and stuff that we went over in lab specifically.
This is a real bone, so you can see that
Okay.
bone kind of broke away here.
Thank you.
All right. I'll
Have a
see
good
you.
rest of your day.
I'll sign on at 3. I'll send you the link and I'll update the announcements to let everybody know the PBL change. All right.
Thank you.
Okay. And sorry, I'm wearing sweats, but I keep jumping out to do the shoveling like all the rest of you too. So it's a fun day, isn't it?
Yep.
That is so heavy. That snow out there is so full of water. I was dying last night when I was doing it.
Gotta
So-
love Michigan,
Would be on the same